So we're now moving on to the 1990s. So let's see what was happening in that decade.
Another turning point, a fork stuck in the road. Time grabs you by the wrist, directs you where to go. So make the best of this test and don't ask why. It's not a question but a lesson learned in time. It's something unpredictable, but in the end is right. I hope you had the time of your life. So take the photographs and still frames in your mind, hanging on a shelf in good health and good time. Tattoos and memories and dead skin on trial. For what it's worth, it was worth all the while. It's something unpredictable, but in the end is right. I hope you had the time of your life. But in the end it's right. I hope you had the time of your life. It's something unpredictable, but in the end is right. I hope you had the time of your life.
So now we're going to hear talks from two of the stars from that period. Professor Richard Troughton, a cardiologist who specialises in cardiac imaging, and Professor Chris Charles, a physiologist and expert in preclinical models of heart disease. Take it away, Richard, please.
Thanks, Saki. Kia ora koutou. It's certainly a huge honour and privilege to be part of this incredible group. And one of the areas that the CHI has led in is the whole concept of biomarker-guided treatment, particularly for heart failure. I think it's a real example of the brilliant basic science translating to clinical utility. And having available an assay for NT-proBNP, which allowed us to get insights into cardiac function, was really one of the basis for the rationale for the NP-guided strategy. As has been pointed out, and we've been able to, and others have shown that NT-proBNP levels are related to cardiac function, loosely related to ejection fraction, but if you look at carefully characterising cardiac function, then we could show that in heart failure patients about 80% of NT-proBNP variability could be explained by cardiac function. So as Eric has shown very elegantly, NT-proBNP is also a validated diagnostic marker, and this is important. Also, the data he showed in terms of response to therapy in that initial cohort. This was further data supported from a large cohort that Mark and John Lanchbury put together, forming the ICON collaboration, which was part of the real validation of NT-proBNP as a diagnostic marker, showing that you could rule out heart failure with low levels, but that higher levels were indicative of acute heart failure, which is important when you're guiding patients because persisting high levels could indicate persisting congestion and risk for heart failure. As well, NT-proBNP is a powerful prognostic marker. Mark had shown this in the MI cohort. But in acute heart failure, it's evident that the change that Eric alluded to with treatment is important. So if you look at changes over time in terms of BNP or NT-proBNP levels, patients whose levels fall had a much superior outcome in terms of 2-year mortality. whereas those patients whose levels rose or remained high had very high levels of mortality at 2 years. So being able to drive down NT-proBNP levels seemed to be a good thing. And it turned out that most of the proven therapies for heart failure, the ones that modified outcome, actually produce a fall in NT-proBNP levels. This was eloquently described in this paper led by Gary, which shows that either short-term or long-term NT-proBNP levels tend to fall if you use proven therapies. The slight caveat was beta blocker therapy, which in the first instance, in the early days or weeks, tends to cause a brief rise. And this was a study that Mark Davis and Mark Richards led, which showed that for patients who were started on metoprolol, a proven heart failure medication, by 6 weeks there was a near doubling in the level of NT-proBNP or BNP. But longer term levels fall and follow beneficial remodelling. So that rationale led to a hypothesis that was generated by Gary, Eric and Mark, and part of the program application in the 90s. The hypothesis that heart failure treatment could be guided by BNP or NT-proBNP levels, and if we could drive down NT-proBNP levels, that could improve outcomes. So the initial study that we evaluated that in was performed in the late 90s. We recruited 69 patients who had proven heart failure and randomised them either to usual clinical care using usual clinical acumen and up-titrating medications according to clinical criteria, or using an NT-proBNP target, trying to get patients below a target. So patients were seen regularly, and this involved Gary sitting in many clinics with me in a blinded fashion. He was blinded to patient group, but he assisted patients clinically, and then I was unblinded and could up-titrate for the NP group because I knew the NP levels. So we followed patients and up-titrated therapy according to group. Mark adjudicated all the clinical outcomes, and at the end of that we were able to demonstrate that using an NT-proBNP guided strategy, we could actually drive down the NT-proBNP levels. So in the NP group, levels were lower by the end of follow-up. But more importantly, we saw a much lower event rate in patients in the NP guided group. So patients were much less likely to have a heart failure event, to be hospitalised or to die during the first 6 months. So that was really exciting pilot data, and it led on to a bigger study, the famously named Battle-Scarred study, named by Mark, and designed by Mark and John and Gary, and led by John Lanchbury, which went on to look in a bigger cohort at NP-guided care, particularly comparing intensive care with usual care versus NT-proBNP guided care, and also looking at the effect of age on that, the effectiveness of the strategy. This again, it received HRC funding and Heart Foundation funding and assay support from Roche Diagnostics. A slightly more complex design and a bigger study, so 364 patients randomised either to usual care, and this group actually we didn't see again really, they were seen in the community or in the hospital clinics, or the intensive group, which either had usual intensively clinically guided treatment or NT-proBNP guided treatment. And we looked at events over 2 or 3 years, so a much larger study, more complex. But at the end of it, the study demonstrated that in young patients below the age of 75, there was a substantial survival benefit. So this is all-cause mortality, much lower in patients who had an NT-proBNP guided strategy compared to either clinically guided care or the usual care in the community. Above 75, their strategy doesn't seem to be effective. So our research really generated a huge amount of interest globally and led to a large number of small and moderate-sized studies performed in Europe and North America. And we had the privilege of being able to lead a collaboration with each of these groups and to combine the data into an individual patient meta-analysis. Chris Frampton worked some statistical magic, as he always does, and was able to help us analyse all the individual patient data. And what that study showed amongst well over 2,500 patients was that this NP-guided strategy seemed to be highly beneficial. So for all-cause mortality, and near 40% reduction in the hazards ratio. But again, the benefits seemed to be largely in the younger patient group, with no effect really overlapping hazards in the older group of patients. We could also demonstrate that there was reduced hospitalisation risk for NP-guided therapy. So as a consequence of this, international guidelines have included monitoring of heart failure with NT-proBNP or BNP as a recommended strategy. So in summary, CHI has pioneered biomarker-guided treatment of heart failure. Current guidelines now reflect that and recommend NT-proBNP monitoring of heart failure and cite our research and our collaborations. And I haven't discussed it now, but the Imperative study has been a follow-on, and in collaboration with our good friend and collaborator Rob Doughty, we've been testing the strategy in acute heart failure setting.
So I actually just squeezed into the end of the 80s, but we'll talk mainly about some of the work that myself and Miriam did in the 90s, and Tim has actually already introduced this a little. And that is looking at NEP or neprilysin inhibitors in preclinical models or sheep studies, basically paralleling clinical studies. And before we do that, so my very first publication was, what is the physiological role of AMP? And actually Mark has already presented studies that showed what it was when they took it themselves as volunteer humans. But we did very similar studies in sheep too. Sheep have advantages that you can do some things that are not ethical to do in humans, but you can also potentially, for example, in this study, infuse for a longer period, 24 hours, or other studies we went longer. And I won't dwell on this, but basically this is very similar to results that Mark showed, show that subtle increments in ANP cause the expected and good reductions in pressures around the body, and also an increase natriuresis. And Eric's also showed and dwelled on a version of this study too, this diagram too, so I'm not going to dwell on that. But basically, we were able to show that sheep are good models of human heart health and disease. ANP and BNP are physiologically relevant hormones that contribute to the day-to-day control of blood pressure and salt and water balance. And our collaborative approach, physiologists working in partnership with clinicians, biochemists, assay and laboratory scientists, makes for a world-class research group with the ability to unravel the role of heart hormones. So just a wee sort of digression. Heart failure, there's actually many hormonal systems which are activated in the setting of heart failure. And basically you can sum this up as the baddies on the left, the constrictors, and the goodies on the right, the dilators. And in severe heart failure, the baddies outweigh the effects of the good systems. And Gary has talked about ACE inhibitors, the very first patient that received one. So clinicians and scientists started realising that if you can inhibit the effects of the bad hormone systems by giving drugs that inhibit those, that you show benefit in heart failure. So through the 80s, 90s and 2000s, there were classes of drugs which improved the outcome, the daily living conditions of patients without heart failure, but also mortality. And you can see each time you introduce one of the new classes of inhibitors, you improve annual mortality. But after the 2000s, it wasn't quite sure whether there would be the next big class of drugs. So during the 90s, we and others around the world started wondering whether we could promote the actions of the natriuretic peptides in the body. Mark's already shown this slide too, so I won't dwell on it, but it's just to introduce that we have a pacing model of heart failure, and Miriam is the person who heads up those studies. But both myself in normal sheep, Miriam in heart-failed sheep, and actually Mark in normal humans and a range of human patient cohorts, we spent a lot of time, particularly in the early 90s, administering these NEP or neprilysin inhibitors to either our animals or patients or human volunteers. So we are blocking the enzyme which cleaves ANP and inactivates it. So ANP will hang around in the body for longer to have a beneficial effect. I'm not going to go in detail through the studies, but I'm going to show you two in the heart-failed sheep. So if you give the NEP inhibitors, given at this time point 0 and infused for a 3-hour period, you can promote the circulating levels of the natriuretic peptides. You can show a good array of hemodynamic or blood pressure lowering and heart improving actions, and you can promote the increase of urine volume and urine sodium excretions. So you're having things that benefit people or animals, in this case, with heart failure. So one of the next studies that Miriam did was she combined NEP inhibition with the ACE inhibition, which was that's the one that Gary talked about right at the start. Standard therapy, if you add the NEP inhibition to it, do you get added benefit over and above? And I'm not going into detail, but basically you do get an additive over and above improvements in hemodynamics and in the urine output and sodium. So smart drug companies around the world started to produce compounds that would combine both ACE inhibition and NEP inhibition. One of those compounds was omapatrolat. We did a sheep study with that here in Christchurch. We also did a human study with that in Christchurch as well. But then soon after 2002, a negative study actually using this compound in a very large cohort of nearly 6,000 patients actually showed that that compound, omapatrolat, had nil benefit over ACE inhibitors alone, and there were also clinical complications with it as well. So at that stage, multiple pharma companies pulled out of NEP inhibitor development, and we all wondered really, was NEP inhibition destined for the big pharma scrap heap? But 12 years later, only 6 years ago now, the Paradigm study, one company persisted, and the Paradigm heart failure study, which had been going for a number of years, was published in the New England Journal of Medicine. And that was a combined inhibitor of, slightly different to the ACE inhibitors, but still inhibiting the angiotensin system and neprilysin inhibition. And although we didn't appear on the author list of this multinational study, of the 28 references that were referenced in that publication, 2 of them were our very own sheep studies. So, you know, we didn't get the final glory, but we contributed some 15 years earlier to the science that is the background to this. And in short, not going into detail, but in that study there were 8,400 patients that were randomised, heart failure patients, to receive either an ACE inhibitor alone or the new compound, and there was a 20% reduction in death and or hospitalisation compared to the ACE inhibitor alone. This compound received fast-track FDA approval, and Entresto it is now called, and it was the first change to heart failure pharmacotherapy in over a decade. And so we're quite proud that CHI preclinical studies provide a key link to the translation of biodiscovery to new treatments for cardiovascular disease. And not in the 90s, but just looking ahead a wee bit more, this is the neprilysin that I talked about before, but this is above the line is on the outside of the cells in our body, and below the line is what happens on the inside of the cells. So once the natriuretic peptides activate their receptor, they increase the cyclic GMP. We've been looking, well, particularly Miriam and Nick, who has now joined the team, switched over from the Omics group about 5 years ago, at inhibiting the degradation of PDE9 inhibitors, and published in a very prestigious journal about 2 years ago, showing that that is active. And we just got an email just a couple of days ago to say, coming over from being synthesised in Australia is osteocrine, which is maybe the next big thing in this area which we will be putting into sheep in the new year. So thank you.